Mackenzie, and my project this year was the role of microglia in white matter injury. White matter is part of the central nervous system. It consists of neuronal processes, axons, as well as myelin sheaths. White matter injury is common in many hypoxic events, such as lack of oxygen during childbirth. There's currently no treatment for white matter injury. So our lab looks at the microglial cell, which is the macrophage of the brain, and it's involved in synaptic pruning, as well as it's a scavenger for different pathogens. It has many pro-inflammatory effects, as well as anti-inflammatory effects that are constantly competing with one another while trying to maintain homeostasis within the brain. And we believe it's the prolonged pro-inflammatory effects of microglial cells that contribute to white matter injury. So in my project, I stimulated microglial cells with MOG, which is myelin oligodendrocyte glycoprotein. This is a glycoprotein that is released during white matter injury. We were also interested in NADPH oxidase, as well as poly-ADP ribose polymerase 1, or PARP1. So I'll explain that in the next slide. So this is a little schematic just to show what happens during white matter injury. So during white matter injury, there's myelin degradation, and this releases MOG, the glycoprotein. This glycoprotein then activates microglial cells, and when microglial cells are activated, the enzyme NADPH oxidase, or NOx, becomes activated as well. Now, NOx is an enzyme that mediates reactive oxygen species production, and this causes activated or oxidative stress, which we believe contributes to white matter injury and increases white matter injury. The other enzyme we're looking at is PARP1, and this is an enzyme that affects the different anti-inflammatory cytokines and pro-inflammatory cytokines secreted by microglial cells. So this leads to our hypothesis that MOG, when microglial cells are stimulated with MOG, it induces activation and pro-inflammatory effects that contribute to white matter injury, as well as when we looked at NADPH oxidase and PARP1, we believe they mediate these effects, and if we inhibit them, they could serve as a potential therapeutic approach to white matter injury. So the first thing we did was we established a dosage curve of the amount of MOG we want to stimulate our cells with. So what we found was 3 micrograms per mil was the perfect amount, as it increased the amount of amoeboid microglia from around 20% to 60%, indicating the microglial cells were activated. And anything above that wasn't a huge increase, so we stuck with 3 micrograms per mil. It also increased microglia proliferation, which is an indicator of the pro-inflammatory phenotype of a microglial cell, indicating it's activated. So there are 2 indications there. So just to show you what an activated microglia looks like and how we can tell, on the left is, you can see they have small central somas with long processes, and they're very ramified. So that's just treated with MEM, which is just a general media. However, when we stimulate these microglial cells with MOG, they become activated, and they're no longer resting, as you can see, through the large amoeboid-type shaped cells with lack of ramifications. So we use BSA as just a control to make sure it wasn't a protein being added to these cells that was initiating the activation of the microglia cells. And as you can see, they're not much different compared to the control. However, MOG did induce a robust activation within these cells. So we want to see what effect of NOx was on white matter injury. So when we treated microglial cells with MOG, you can see there is an increase in amoeboid microglia. However, when we used NADPH oxidase inhibitor known as apocyanine, there was a dramatic decrease in the percent of activated microglial cells, indicating that NADPH oxidase had a role in the activation of microglial cells and ultimately white matter injury. When we looked at proliferation, there was also a decrease in proliferation when the cells were stimulated with MOG and apocyanine, the NADPH oxidase inhibitor. So next we want to look at superoxide production. So on the right-hand side is the progression diagram of superoxide production over 90 minutes of the microglial cells. And around 75 minutes, you can see a dramatic peak of the cells treated with MOG. And when the cells are treated with MOG and apocyanine, there's a large reduction in the amount of superoxide compared to the ones treated with MOG only. And on the left-hand side, it just quantifies this data during the 90-minute progression, just focusing on the peak time of around 75 minutes. So one other factor that indicates microglia are activated and in a pro-inflammatory state are cytokines. So there are pro-inflammatory cytokines as well as anti-inflammatory cytokines. And when microglia cells are stimulated with MOG, there's an increase in pro-inflammatory cytokines. So interferon gamma, IL-1s, as well as TNF-alpha are pro-inflammatory cytokines, and they were increased when stimulated with MOG. A very interesting thing, though, is when they were treated with MOG and ampicillin, the pro-inflammatory cytokines decreased, and there's actually an increase in anti-inflammatory cytokines, such as IL-4. So it shows that the microglia cells were switching from the detrimental pro-inflammatory state to a more anti-inflammatory state or phenotype. So currently we are looking at part 1 inhibition. So part 1 is an enzyme that controls different cytokines that are released by microglial cells, and it's a factor on the activation of microglial cells. So I have looked at the morphology so far with these microglial cells treated with a part 1 inhibitor known as olaparib, and we have found that it also does decrease the percent of activation within these microglial cells compared to the controls of the assay. So the work in progress, we currently have cytokine and trophic factor samples ready to be sent off for analysis for cytokine. We also still have to look at the proliferation and superoxide production within the samples as well. We just didn't have time, or haven't done it yet in time for this presentation. And I've just finished optimizing the oxygen glucose deprivation and retribution experiments. So this is more of a physiological representation of white matter injury, where we insert microglial cells into a hypoxia chamber and starve them of oxygen and glucose for 60 minutes, and then reperfuse them back with glucose and oxygen. And we're going to compare these to control groups, which are kept in an incubator with the proper dosage of oxygen as well as glucose, and see the effects on this. So that is in the works right now. So just to conclude, we found that microglial cells, when stimulated with MOG, induced activation in the morphology, proliferation, superoxide production, as well as cytokine production. However, when we used NADPH oxidase inhibitor, we found that the pro-inflammatory state was reversed almost to an anti-inflammatory state. Proliferation was decreased, morphological activation was decreased, and anti-inflammatory cytokines were increasing. So we are currently also looking at the part 1. So far we see that there's a reduction in morphological activation, and yeah, the rest is being continued with the part 1 inhibition. So I'd like to thank my supervisor, Tiina Kauppinen, as well as the tech, Gary O'Dara.
I have a question about MOG, and maybe I kind of missed it, but like, how is it active? Is it getting into the microglia, or is there like a subsurface receptor that binds?
Well, microglia just become activated when they're in the near vicinity or even when touching whatever stimuli, so it's released, and I'm not sure if it's a receptor or not, but microglia cells are very sensitive and they sense any difference within their environment. So.
How do they sense the environmental changes?
Immune cells don't know the difference except always just usually anything that they consider as a pathogen, if they activate, there might be something said that that's not going to happen.
Great talk. I just wonder, how long is the anti-inflammatory phenotype around for? Like, do those cells stay there permanently? Or you guys have done any time studies to see if maybe they switch back eventually to the pro-inflammatory stage?
Oh, with the like the morphology and stuff.
Mm-hmm.
We just look up to 24 hours right now, so I don't know if it ever go back if you didn't keep applying a lot further or not, but that was fine. But yeah, we just fix them after 24 hours. So.
This MOG for vitals, is it used in clinical applications at all, or is that?
So